Okay, good afternoon. Start again. It's a real pleasure to be here and to speak to you. I'm not going to assume that you necessarily know too much about mass spectrometry, so I'm going to start from quite a basic level, tell you a little bit about the history of where we started and very recent results of ejecting complexes directly from the native environments. And then if there's time, I thought I'd show you. I really enjoyed the sort of worked examples this morning of EM, and I thought I'd show you a worked example of really complicated assignment of mass spectrometry so that you can sort of compare the processes. So I'm going to talk about membrane protein assemblies, including GPCRs, and then move away from detergents into native-like environments, and also a little bit about where I think the field is going at the end. So tribute to my great mentor, Chris Dobson, who sadly passed away last year, and I know he was a great friend of many people in Israel, so just to your attention. And he really started my career really encouraging me to be very bold with experiments, and folding with mass spectrometry was one of those. That was not possible at the time. It was not really considered a feasible experiment, but we were able to show through hydrogen deuterium exchange labeling that we could preserve the folded state. And this was also with Sheena. And this is the folding of hen lysozyme. We've moved on quite some way from there. And the next question, of course, if you're folding an individual protein molecule, is to start to think about how cofactors might accelerate folding. And this was an experiment that I was still doing with my own hands. And we thought about the folding of a very simple 4-helical protein. And would it fold faster if we templated it with its cofactor, in this case a CoA? And I was sitting at the mass spectrometer, and I often tell students that there are days that stick in your mind, and this was one of them, because I was folding, and I saw folding, and then I saw the complex, and this was of the protein with the CoA ligand bound. And everything I knew about folding was it's a solution thing. If things don't stay together in the gas phase because there's not the water around them to keep them stable. But I saw it, and so it's that very unprepossessing peak which changed my career, really. It doesn't look particularly good. There it is. That is the mass of the two things bound together. And at first we thought, well, maybe we should tune the conditions. Maybe we don't want to see these cofactors binding. Let's see if we can get rid of it. And then we realized that actually it was obeying everything we knew about the protein and the ligand. If we put in a ligand with a longer tail and a more tighter binding, or if we mutated some of the critical residues around here, we could change the binding, and it reflected what we were seeing. So we started to believe that maybe this was telling us something about protein folding and structure in the gas phase of a mass spectrometer. This is a very small system. I got very excited about this, and with my first grant I said I want to build a new mass spectrometer that will go to much, much higher mass to charge than we've seen before. I'm going to put all these amazing assemblies in. It was very hard to get funded, I have to tell you, in that climate. But we managed to get funding. We managed to get some people to help us to replace the quadrupole, the collision cell, and all of the different parts. And actually, Mikal Sharon, she used this in my Cambridge laboratory when we got this set up. So I'm very proud of that instrument, and unfortunately it's now been scrapped. But it was a wonderful thing to have in the lab at the time. And this, again, was one of the first things I wanted to do. We'd looked at individual proteins. Could we really do an assembly? And at that time, I wasn't very good at preparing proteins, so I went for something that was naturally expressed in plasma and purified this transthyretin retinol binding protein complex. This was my first independent paper, and I decided to publish it in a journal that no one had really heard of, which I think is quite interesting now. It gets very few cites because that journal's subsequently been renamed and relaunched and a million times over. So I think there's a message there if you should be a bit more bold, because I said something like, well, maybe this will be general for protein analysis, and then I spent the next 20-odd years trying to prove that it actually does work across a whole range of different systems. And I couldn't come to a meeting that Venki's invited me to without mentioning the ribosome. So this was one of our early successes on the new instrument in Cambridge. Just really, we were very interested to see how far in mass and complexity we could go. And you might think this looks a bit ridiculous, which I do too, but this is modelled on, but it showed that there were actually, in this particular Thermus phaemophilus strain of ribosomes, there were actually 6 L7, L12, and this was borne out by crystallography of just this part. So we just docked it on. We don't imagine for one moment it looks like that. But one of the things that, of course, you can do with mass spectrometry is really study very dynamic, very heterogeneous assemblies. And this, there's phosphorylation going on, and there's all sorts of folding and unfolding. So it's not adverse to looking at very dynamic molecules. I used to work out the chemical formula. This is really showing my pedigree of something of a mass of 2.3 million that we could measure, plus or minus 2,000. So I worked out all the carbon, hydrogen, and nitrogens at one stage. But it's really, you can't measure something of that size with sufficient accuracy, and that's an important point to make. And here's the 30S. This is this stalk, which comes off as an independent species, and this is the tRNA. But of course, you can show that you can maintain something as large as this, but there was always this question of what shape. How do you know it looks anything like a ribosome? It could be completely unfolded. I didn't ever believe that it was, because it bound factors and it behaved as though it was a folded molecule. We then collaborated with Waters to look at the collision cross-sections of these molecules. So collision cross-section is a measure of something as it goes now through here. You'll see that it gets buffeted because we've got a counter current of gas against it, tumbling away, artistic license coming towards you, and then measured in the normal way in our time of flight. And if you look at this region here, this is the part that can separate molecules according to shape. So, of course, you have mass to charge, that's one property, but folded and unfolded have the same mass to charge but different shapes. So we could now add a new dimension to our measurements. And this is work of a husband and wife team in my lab, Brandon and Maggie. They weren't married at the time, but obviously the project went well and decided to stay together. The complex that we chose was actually a very fortunate one. It's the trap RNA complex. It's the 11-mer, and it has 2 different conformations, a ball shape and a ring shape. Same mass to charge, readily separated. So we could show that these complexes, if we treated them in a certain way, recapitulated the sort of shapes we would expect. But there was one more thing that sort of haunted me. Get to the end of every talk and somebody would say, well, why can't you do membrane proteins? It's in the gas phase, surely it doesn't matter about solubility. If you're in the gas phase, you're free of all the restrictions that hamper people in solution. And of course, that's a very attractive thing, but you've got to take them first from solution into the gas phase of the mass spectrometer. And we struggled with this for many years. And this actually first happened in the Cambridge basement. There's a lot of good things happen in there. Where there's no daylight, and it's not great, but where somebody decided that actually what you need to do is just make a sort of giant encapsulated bubble to take your complex from solution into gas phase, and then once you're in the gas phase, get rid of the outside of the bubble, which is the detergent. So if I show you, this is what it looks like if you were sitting in front of the mass spec, trying quickly to tune it. You'll see you're ramping up your collision voltage here. You're getting out these very nice peaks. And if you overshoot, go too high, it starts to decompose because you've released a subunit. If you get it just right, at around about here, it's intact. And then if you don't have enough energy, you just get this really ugly, I think somebody this morning said junk, and that's kind of what it is. It's junk. It's detergent, it's lipids, it's unresolved chemical stuff. We don't really know what it is. But this is what we magically do. We shake off all the detergent and just keep the complex in the centre. I was very fortunate also in Cambridge to work with Daniela Stock on her thermos thermophilus ATPase, and this was a beautiful thing for mass spectrometry and also for her. We got a really stunning, I think, even by today's standard, intact mass spectrum of this thermos thermophilus ATPase. And she was in Australia, and I was in Cambridge, and we used to email late at night discussing what we thought was the subunit stoichiometry, because at that time there wasn't any high-resolution structures, and we weren't sure how many L subunits there would be, how many of these arms there would be, and whether or not there were any nucleotides or lipids associated. This took a long time to assign, but eventually we finished with that, and we came up with a model whereby we actually had 12 subunits, but in the presence of lipids, we reckoned we have, we know we have 6 lipids, which we think actually changes the conformation of this ring, taking it from proton pumping to ATP synthase. There's only one ATPase in thermos thermophilus. But this got us to think about this, because there were 6 lipids here, and in a different one we did different numbers of lipids. These lipids were clearly having a regulatory role. So it made us think, well, how are we going to distinguish whether lipids are important or just coming along for the ride? And this is the work of Kalur Gupta. He's now a professor at Yale. And he, assistant professor, I think they call it, he decided to look in the PDB, come up with a whole range of membrane proteins that have been crystallized, and ask the question, are they stable in the absence of lipids? Are there lipids in their structures? Can we see the lipids in our mass spectrometry? So we made an awful lot of these, and we looked at their mass spectra, and some of the ones that you think might require lipids, such as the ligand-gated ion channel, it was perfectly stable without lipids. Similarly, water channel, AQ2Z, and the ammonia channel. But there were some in this corner here which had a very low number of salt bridges, that's denoted in black, and very little buried surface area. So if you think of a dimer that's sort of end on, and it might require then lipids to mediate its interactions, whereas if you have a dimer like that with masses of buried surface area, perhaps lipids would not be needed. That was the hypothesis. And then we made a few of these in this corner here that looked particularly interesting. We found that the leucine transporter, which is the prokaryotic version of the dopamine transporter, this definitely required lipids for dimerization. And then we chose 2 sodium proton antiporters, one from a thermophilic species and one from E. coli, reasoning that this one was sufficiently stable to not require lipids, whereas this one would need lipids. And this was actually really nice because it's one of those very clear experiments. So on the right-hand side is the thermophilic sodium proton antiporter, and you'll get used to my mass spectra by the end of the talk, but this is a beautiful one. 10 protons, 11, 12, 13. So from here you calculate the molecular weight, but you see that these peaks are essentially naked. There's nothing else coming with them, just the pure protein, and it's a dimer. So the mass here is a dimer. If we look at this one, you can see it's almost the same sequence, except clearly there's a lot more things going on here. There's a lot of lipid binding that we can see deliberately. I should say that they were both expressed in the same membrane and treated to exactly the same isolation procedures. So it made us think that perhaps there's a predictive scale for lipid dependence, so that if you have, well, we called it delta G dissociation here, but reviewers asked us to change that. So anyway, it's now based on buried surface area and salt bridge interactions. This one is the thermophilic NAPE. This is the E. coli version, much weaker, and you can see almost that there's a cavity here which is filled in the thermophilic species. Semi-sweet I didn't talk about, and Lu-T. Actually, sitting in my office, and I thought, oh, that looks just like what I've been seeing for GPCRs, where people say, oh, maybe they come together, maybe they don't, maybe they have actions in the membrane. So I started to think about GPCRs, and I looked at the ones that had been crystallized, and it was quite interesting. This one wasn't thought to be particularly interesting because it was thought to be a crystallographic feature, whereas these were thought that maybe they were coming together transiently, either for action or for other reasons. And you can see here they've even started to model a bit of lipid density. So we thought, let's have a look at some of these. Let's see if we can explore the whole lipid-mediated dimerizations in GPCRs. That was our question. But we actually went off and found something completely different, as often happens in science. And Jason from Taiwan is the hero here because he worked very hard to get this pentameric complex. So this is the adenosine GPCR, A2AR, binding to mini-GS that we have from Chris Tate, a nanobody, and then gamma and beta to form this pentameric complex. And he worked, I would say, day and night to get this to work, very dedicated, explored all sorts of different detergent combinations, detergents and lipids, our homemade detergents, all sorts of things came into play. And he managed to get this spectrum. And I remember he sent me an email saying, I've got it, it's amazing. And then I saw it and I was really disappointed because it doesn't look so great. You know, when I showed you the previous spectra, they're all sharp and nice. And here, this is what we call noise in our world. This is not particularly great. But then if you isolate just a charge state here and break it, you can see very beautifully you've got all of the components that you wanted. So the A2AR, the nanobody, trimer, other trimer, dimer, etc. So all of the building blocks are there. But what was interesting was that the receptor came out with 2 lipids. So suggesting that in this coupling complex, lipids were present. So we were very excited about that. We found out that it was PIP variants. And we looked at then all possible PIPs, because actually with mass spectrometry, people often say it's all about charge complementation. It's nothing real. You know, we don't really believe in it. So I thought, okay, I'll take one that's even more charged, and if that doesn't bind as well, then it'll demonstrate. So PIP2 bound much better than PIP3, which actually has more charge, and PIP is in the middle there. So this is definitely the one that the receptor prefers. We can then do all sorts of MD simulation and site-directed mutagenesis. So we identified by MD all the potential PIP2 binding sites, and then we did lots of mutagenesis. And this TM4 region turned out to be a real hotspot. And we were really pleased with that because that is the interesting part of the molecule. So as you know, receptors bind to agonists, which bind to this trimeric G complex. And around this region, we thought PIP2 could be having a signaling effect on downstream coupling. We were very happy to collaborate with Chris, who gave us some of his mini-G. This is his structure of mini-G bound to the A2AR. And then what we did was to take the receptor, the mini-G, and then challenge it to bind PIP2. And I know these are complicated. Chris told me people would only look at this and not these. But the data, I think, are really quite amazing because they're very well resolved. So in the top case, I'm just showing you 1 PIP. This is in the absence of G binding. So 1 PIP is the prevalent peak. Once you've got mini-G here, you can see that it really takes to PIP2. So it really acts to bind very selectively and to recruit PIP2 around it. So we realized that we had a way of measuring this, so we could now go off and look at the effects of all sorts of things on the whole GPCR field. We're extremely excited to look at orthosteric, allosteric, agonists, modifications, because of course this gets phosphorylated, and then all of these different Gs have different masses, so the whole combinatorial effect of looking at these became very exciting. So here's just a few examples. So orange means formed a full complex with the downstream, its cognate downstream G. In this case it's GS, but we've shown that PIP2 actually enhances the selectivity here. And then this is only a partial agonist, and you see we've got much less complex. than we expected. And the other thing that we can do, and I'll talk to you a little bit more about that in a moment, is take receptors where we don't know what the ligand is and challenge those in the mass spectrometer and break them down and work out perhaps what they might be doing. Because there's an awful lot of GPCRs where nobody knows quite what they're doing. But we caught the eye of investors at this time because people said, oh, you've got a new way of finding drugs for a drug discovery platform, because you can read out directly whether it's an agonist, an antagonist, whether it promotes GQ, GS, GI. And we realized quite quickly that we were not going to be doing this in the lab. It took us about a week each on each of these, and they want to do 100,000 molecules. So we spun this out of the company into a company on Science Park to do high-throughput screening, and that's going fine, that's going well, but it's not something you can make PhD students or postdocs do, I don't think. Okay, so that's the first part. We summarized this with this PIP2 bridging, which really does have this selective G protein coupling. And it was quite interesting. At a conference, I met with Brian Kabilka's group, and they said, oh, yeah, we read that paper, but we didn't really believe it. We thought it was probably a mass spec thing. And now it's actually not just come out, but he highlighted it to me that they'd actually found PIP2 exactly bound where we'd suggested in the high-resolution EM structure of the neurotensin receptor coupling to the beta-1, to the beta-arrestin complex. So I was really happy that he remembered that conversation where he said he didn't believe it and found it in his structure. So I'm very pleased about that. But it also made us think a lot about the effects of detergents on subunit stoichiometry and on lipid binding. Are we actually forcing some of these because we're choosing certain detergents, or is that how it is in the membrane? And also, if you look at structures, they seem to evolve with time. And so I'm very interested in the patched receptor, and this was a dimer-like, and actually this was monomeric, then it became a dimer, and then it became a tetramer. And these were all under different conditions. Both of these were in Ning Yan's lab, and this is in Egon Shea, I forget which lab that was from now, but they just seem to be evolving with time. The BAM complex has different stoichiometries depending on how it's treated, and the TON complex always has also has a variable stoichiometry. It tends to change a bit depending on its detergent. So we decided that if we wanted to get a handle on this, we should try to go directly from native membrane environments. And this is the work of Dror Chorev, who came from the Weizmann, from Mikhail Sharon's lab. And we were interested to see if we could take vesicles. So these are vesicles that form, which have the natural membrane still around them. We would put them into a cell press, apply osmotic shock, and form these sonicated lipid vesicles, and then take those directly into the mass spectrometer. So we're taking something very, very heterogeneous and then ejecting from that with a high voltage, a charged up complex on the surface of the membrane. Taking that through, breaking it, but they still have quite a lot of lipids associated with them, of course. And when we did this, many of the complexes that we know and love were sort of changed. This was helpful with Lindsay Baker, who did all the corresponding EM to see what our structures were looking like. But we saw many of OMPE, for example, with DNA-K. Lots of things came with attendant chaperones, which confused us for a long time. So this is OMPE, which has this leader sequence binding to DNA-K, which has been reported previously, but we weren't really expecting to see that in our particular prep. And then, surprisingly, this BAM complex was more heterogeneous than we'd imagined, because we'd also studied this in a recombinant sense and agreed the 5 stoichiometry. But of course, that's a recombinant complex in which 5 copies of the protein are present on one gene. In the natural environment, there's actually an additional E, and there's this very heterogeneous lipid composition. And you may look at that and think it looks really ugly. One of our reviewers at the time said, oh, you know, these peaks are very broad. But they are broad because if you have a lipid binding, it's not one lipid, it's actually a family of lipids. And this is the different chain lengths of all the cardiolipins that are present in E. coli. So it's not one cardiolipin, it's the whole family of cardiolipins, which is why these peaks are broad. The sharp ones mean that they're protein only. So you can distinguish quite quickly. And Saima, I like to say that she colours her proteins and hair to match, so I think it's a nice thing to do. And she showed that we could actually get up to 3 cardiolipins binding by MD simulation, and that it was possible to dock an additional copy of subunit E. We then went on to E. coli inner membrane, and we found actually, interestingly, some things that actually bridge the 2 membranes had survived in this particular experiment. And we saw an ATP synthase with its sec-YEG complex. We kept seeing this complex that was larger than ATP synthase, and we couldn't work out why. And it's come with its thing that inserts it into the membrane, so very sort of native-like conditions. Lots of other complexes that I won't go through. Yeah, just surprising that these multi-drug exporters survived the process and are still 2 of them attached. And we then maybe went a step too far into bovine inner mitochondrial membranes, and we could see many things. Again, this ANT1, which was controversial, binding to palmitic acid, which is an uncoupling reagent. And I like this because to me it seems like it's caught in real time, so it's a translocator or transporter, and it's not just got one molecule. So if you take something in time, you might have a slew of molecules being transported, and here I can see, with a bit of imagination, up to about 4 of these palmitic acids going through this transporter. Actually, this initially was a bit disappointing, because although we got some beautiful things about the ANT1 and a little bit on complex 3, complex 1 just seemed to be sheared completely by our procedure, and we didn't see any ATP synthase at all. And then we realized that if we kept the outer membrane around the inner membrane, we would have some protection. This is my poor drawing of a mitochondrial membrane. And we could then see this complex 1 here with cardiolipin binding. So this is actually quite, I think, remarkable resolution for something so large, up at 14,000, coming out of a mitochondrial membrane from one cow's heart. Sorry for the cow. This is complex 5, complex 3, and this is complex 1 out of the membrane. You see beautiful lipid binding. In detergent, we get different lipid binding. In fact, we get both the same complex, but you see this one's less charged, so it's higher, and this one has different lipid binding properties, suggesting that we are changing the lipid binding by the detergent we select. This is, we were criticized for this, for it being broken, but actually this is an intermediate that's also been seen many times in EM. Happy to say. Anyway, we decided we were super excited by this new method, because what you're actually doing is you're taking things in the membrane, charging them up, and ejecting them with a sort of high field. And so we thought, right, high field, dramatic electricity going on. They'll love this for science. And they said, no, we chose this. The avocado sandwich, much more appealing. But now where can we take this? And I do think that we have a lot of opportunities to apply this to all sorts of different membranes and complexes that haven't yet really been worked on or discovered, because they're very difficult to study. Solute carriers is one such class because, well, for me, I think they're very context dependent. So if you try to take them out, they just fall apart and lose all their shape. So I think doing them directly from a membrane is really going to help. And this is a collaboration with Katharina Dürr at the SGC. And we're interested in, well, there's thousands to choose from, really. I'll just talk to you about this one. So this is solute carrier 25A13. And first of all, we extracted it in detergent, and this is one of our in-house G1 detergents, which works quite nicely. And it's a dimer, and it's got a lot of lipid binding, 1, 2, 3 cardiolipins. But again, is this something that we're forcing with the detergent, or is it as it is in the membrane? So we then took this directly from a plasma membrane, and you can see, well, I think it's very exciting because you can see there are small molecules binding. I like to think that it's transporting its amino acid, which is its role. You can see 1, 2 there, and then you can see cardiolipin binding here. And then she didn't believe it, so she did it all again and got very similar experiment again, small molecule binding and lipid binding, and this is directly from a plasma membrane. And I'm sure you will maybe have noticed that we got quite a lot of criticism for this work because people say, oh, well, you know, did you just guess everything? And we said, no, we did proteomics, we did lipidomics, we did endless measurements. But it actually, if somebody's critical, it actually improves your science, and you go off and you think, right, we need a new instrument that will really take away any shadow of a doubt as to what we have here. So we now need to introduce yet more stages of mass spectrometry. So you have your protein going in from the membrane, then you select it, then you break it, then you select the bits that came off, and then you break those until you can go down to the very smallest molecule. And this is the work that Idi and Joe have been working on for nearly 3 years, I guess. It's taken a long time to get this working, and it's on a commercial instrument, but it had to be heavily modified. So this is, if you're interested, this is known as the Eclipse. I don't think you're going to buy one, but it's very expensive. It's actually getting ridiculously expensive. It's like £1 million for this piece of instrument now. In goes your membrane protein from the membrane through here, into here, and then you can do this multiple in and out of the mass spec each time to select something, break it, have a look at it, select something else, and kind of keep on going until you've worked out what it is. And I look at it a bit like this. We have this sea of things. We pick out one complex, we look at it, we look at the proteins, then we look at the lipid, then we break the lipid, and then we get the exact structure of the lipid. So this is native mass spectrometry meeting with omic technologies. And so this is not my name, but the guys in my lab decided they wanted a name for this, so they're calling it native omics. And let's see if it catches on. Well, actually, the manufacturers started using it already ahead of our paper, which has only just got accepted because most of the discussion about the paper was about the name. Can you really call it that? So the paper, they decided, I think, that we can keep the name. But this is just really showing why I think it's going to be important to work with EM, because quite often people have this density. This is actually a crystal structure, not EM, but this is the mitochondrial transporter TSPO, and it has this unclearly defined density, and the author fitted this particular lipid, but then there were some more comments in Science where they said, actually, we don't believe that. That could be a detergent molecule. That could be monoolines purified in here. We don't think you fitted the ligand properly. And so the view is then that you could maybe take this complex, release this ligand, break it until you can work out exactly what it is. And that's what we're doing here. We've taken this complex. We've seen that actually this peak that binds to the intact dimer, in this case, corresponds to this lipid here. So we've isolated this lipid and we've broken it and got its different chain length. And if we do that, it's this lipid, and then fit this in here, sit nervously in front of Richard, saying, oh, that's not a good fit. I think it fits much better than the original one, and there's all sorts of complementarities that we can make with the backbone of the protein to show that actually this is the right ligand to fit in this density. So, long story short, that was a proof of concept that we can take this approach and go from a complex to a protein to a lipid to breaking the lipid and then sort of stitching it all back together again. So I'm going to sum up this part, and then I'm going to show you a worked example of what we're doing. So this is my timeline of using mass spectrometry, starting in '92 to look at folding intermediates, peripheral membrane proteins on the surface of membranes, designing these instruments and persuading people to help me to build them. And then really our breakthrough came when we could release things from detergent mice cells, because that led us into the world of membrane proteins. Collision cross-section was important along the way. Identifying, I didn't talk about this story today, but identifying lipids that stabilize a complex by using unfolding. So rather like guanidinium unfolding, collision cross-section unfolding to tell you which lipids are important for stability. ATPases, and actually right at the end is GPCRs. And you might think, well, why did it take so long? And that's because, again, if you take them out of the membrane, there's not much to support them. They're very floppy, very hard to get the drugs and the lipids to stay bound. And then very recently, excited by the mitochondrial opportunities. And then as I said, I would like to tell you now to something that's not yet published, and this is to do with an ATPase, which is actually in the synapse. And it's this ATPase here that helps load the neurotransmitter across the synapse. So it's kind of, John Rubinstein contacted me and said, oh, I've got 2 rat brains, I've isolated some of this prep. We don't know which isoforms are present, we don't know how many copies of any of the proteins, we don't know what cofactors are there, and we've only got 2 brains worth of material. And I thought, it's never going to work. So this is really a test case for us, and it worked very nicely in parallel with his EM. So he gave us the preparation, and what I wanted to show you was a typical workflow, but I think that might be a bit boring, so I'm not going to show too much. But first of all, you would start with all the proteomics that you could do, all the lipidomics that you could do, so that you've really characterized as much as you possibly can. So you've got all the masses of all the pieces that are possible. And then this is the spectrum. And I show this because, as I say, people think, oh, maybe they just guess what things are. And of course we don't. We spend a long time looking at these. And for me, this is like a 3-dimensional puzzle that will engross me for many hours. And then I look up and it's time to go home. But this is how it looked. This is the first spectrum at 100. So this is 100 volts on a particular cell. So 5 micrograms isolated from synaptic vesicles. And you can see that there's resolution, and some other proteins have fallen off or are coming along for the ride, as we say. And as we increase the energy, this area of the spectrum gets higher, and that's because these are stripped complexes. So you have your intact, you remove a protein, and the residual bit sort of goes in asymmetric pieces like this. So here's your intact, a protein comes off, you form a product that goes up, etc., etc. And this is incredibly complex. So you sit there with all sorts of possibilities, and of course we've got programs and software to help us to assign. If you now expand this region, you can see, hopefully, that this is really quite nicely resolved. We can start to pick series of peaks. So series of peaks means a particular complex. So this is one complex. And now what we're looking for is different isoforms incorporated into the ATPase. So the question is, can you distinguish the brain ATPase from the synaptic ATPase? We're expecting, this is when it became ridiculous, we're expecting a difference of about 20 Daltons. So what, you expect me to tell that within this forest of peaks, but in fact you actually can, because what you then do, so we're looking for G1, which is one isoform, and G2 is a different isoform. So they're about 43 apart. And what we found was 2 series of peaks here. This one was different from this one by 13606. So this was actually a bit disappointing because it wasn't really the mass we were looking for, either of these. So it was between them. But if you look as these products go up, the low mass end comes up. So here no products are coming. Here you start to see products coming. And here you can start to assign proteins. And we only saw G2. So if we go back one, as we lose this mass at the bottom, this one begins to appear. And so this enabled us to distinguish G1 from G2 and to convince the reviewer that this is actually from the synaptic vesicle and not from the brain. And I show you this because I'm very proud of it. I think it's an amazing spectrum from rat brain, 5 micrograms. And look at the sort of heterogeneity of different isoforms and subunits that you can see in here. Predominantly one species. Here's a different species in blue and a different one in green. And in general, we work extremely hard on these for a number of months, and then the editor says, can we put it in the sup? No one will understand it, and it's so disheartening. But this time she said, I'd like to put that as figure 1. So, oh, thank you. But of course, everybody wants to see this one, which is much more beautiful. But all of this was confirmed by proteomics, lots of different subcomplex formation. And it has to, of course, be rigorously tested in all different fragmentation pathways and lots of different programs. So it's not true that we just guess our assignments. Okay. With many, many thanks to an amazing group of people, I realised when I was putting this in this morning that the building looks great, but you can't really see anybody in the picture, so it's a bit unfortunate, but I didn't mean it that way around. They should be much bigger and the building should be less. But it's been great working with them all and really talented, and I've tried to mention everybody as we go through, all my collaborators and my funders, and thank you so much for your attention. I'm happy to answer any questions.
Great, we have time for a couple of questions, preferably from the audience. But I have 2 as backups, but let me try my first one, Carol. So it's very impressive, and I think you've completely convinced me, at least, of all of the interpretations. What would be very interesting, I think you've thought about this before, is are these proteins that are flying through the vacuum, if you caught them at the end of it, rather than breaking them all up, do you think, would they still be active?
That's very interesting. So we've thought about this a long time, and the first examples of viruses, of course, are active because we all know that we can pass them through atmospheres and they're still active. So certainly for viruses, but I don't really see that as a particularly difficult test case. So we have now been looking at collecting, which we have done from inside the mass spec. Apoferritin looks good, and that again is a robust molecule. So at the moment, that is a very fruitful and active area of research. I personally don't think we'll ever use it to prepare your grids, but I do think we'll be able to look at things, and to mass spec playing more intimately with EM is, I think, a very good way forward.
You don't get enough molecules at the end of it. You get enough to get a peak, but you wouldn't get enough to do an enzyme assay, for example.
No, we would. We would just collect for longer than we typically do. So currently we are collecting molecules, and we're collecting for about an hour, and then we can have enough to do enzyme assays, to do to prove to people that it's still folded. And
it's just interesting. It could be that there's some subtle thing, you know. Yeah. One residue goes like this, and then you have to bring it back. Or, anyway. So,
I will ask another question. Was this ATPase active at all before?
The rat brain synaptic vesicle. Yes. I guess it was in the brain. I didn't do the test. For sure. In the
purification.
Yes. John Rubinstein did all of the assays. I didn't get involved in that side of things. Sorry?
Okay. I didn't see them, but...
No, I didn't put them in to the talk.
Carol,
I was really fascinated with the mitochondria. You have the 2 membranes or bacteria, but I sort of don't quite understand when you have 2 membranes and things are flying out. How do you know they haven't kind of made friends with a protein from a different compartment? I was thinking about URON-P, which is in the bacterial outer membrane, but DNA-K, which is in the cytoplasm.
Yeah.
So how do you know? Yeah, can you get rid of that worry?
Yeah, no, the DNA-K I'm perfectly happy with, and we don't say that they're definitely the right way round. So they can invert, of course, so you can get both sides of the membrane exposed. And I think the chaperones actually act as chargeable handles to help pull out the complexes, because quite often we don't see it without its chaperone, and it must be heaps of it there without its chaperone. But I think the mechanism is you have it in the membrane, you need something to pull it. So you have to charge it up either with a chaperone or with ATPase. Of course, it's got a big head, so that charges and pulls it out of the membrane. So I'm quite happy that we're not keeping them as we think they are. So it's not the outer and the inner membrane intact, because we sonicate them and we expose things on either side of the membrane.
Okay, so you've got inside out and also membrane fragments as well,
yes,
have you?
Yes.
Okay, thank you.
So that gives you access to both
So that gives
sides.
you something else to worry about.
Yes,
which
if you want to.
You're very good at worrying, so
well,
we'll
I'm
let
trying
you
not
sort
to
out.
worry. I just think it's exciting, but of course other people worry about it a lot.
Perhaps I can ask one last question
Yes.
then, Carol. Of course, we're all very interested in the cost of these things.
Yes.
And so and you mentioned £1 million, but presumably you are buying it, and the entire risk is taken by you. If you were to have such a commercial machine, and the manufacturers had to deliver it and guarantee it would work, it would cost a lot more. So, how much would it cost if somebody else wanted to get exactly your latest instrument? How much do you think it would cost?
Yeah, I mean, I have been involved in these negotiations because people say, oh, well, Carol only paid $1 million, why have I got to pay 1.5? And I say, well, we spent 3 years developing it, and I still think $1 million's a lot if you spent 3 years developing it, but I couldn't get it any lower. But you don't have to come in with that high-level instrument. You could enter with a different one. I sound like I'm trying to sell my spec, which I'm really not. I don't have commission.
Okay, so, Carol, thank you so much.
Pleasure.